Hello everyone, my name is Széliová and I'm a postdoc in the group of Jürgen Zanghellini at University of Vienna and also at Austrian Center of Industrial Biotechnology. And today I will tell you how we improved the predictions of CHO metabolic model with experimental data. So first I'll start with a short introduction about Chinese hamster ovary or CHO cells. These cells are the most popular cell line for the production of protein biopharmaceuticals, especially monoclonal antibodies. And their main advantage is that they can perform human-like post-translational modifications, especially glycosylation, which is very important for the properties of the biopharmaceutical. However, the cell line development with these cell lines can take several months, and it often involves trial and error approaches, for example, targeted evolution or high throughput screening, and often you need to screen thousands of clones until you find a good producer, and then in the end you don't even know if why it is a good producer. So here constraint-based modeling could hopefully help to make this process faster and more efficient. So in 2016, a genome-scale model of CHO was published. And the question is, if we want to use it, what experimental input do we need to use it successfully, and how important are these inputs for the predictions? So in this work, we focused on the exchange rates of extracellular metabolites, biomass composition, and maintenance energy. So first, I'll start with the biomass composition. So we wanted to know how variable is the composition among different CHO cell lines and conditions, and to see if there are some components that we can generalize for CHO and which components need to always be measured. We measured composition of 13 cell lines and conditions in exponential phase. And here we can see the macromolecular composition. It was very variable. So for example, protein content ranges between 46 to 66%, so quite a range. But we didn't see any consistent differences between producers and non-producers, but we saw some effect of medium. We also measured amino acid composition and lipid composition, and we saw that amino acid composition is constant across all cell lines and conditions, but lipid composition is strongly influenced by medium composition. And if you want to know more about the biomass composition and its variability, you can check out the poster of David Ruckebauer. So then we checked what is the effect of biomass on the predictions by FBA. So here you can see the predictions of growth rate with generic biomass composition that was in the original model and with cell line or condition-specific biomass composition. But you can only see a subset of the cell lines. And this is because we only saw the effect of biomass if we took cell lines on conditions with highly accurate exchange rates. If you would take all the data sets, then we did not really see the improvement. And this showed us that the most important input for modeling is actually the extracellular exchange rates. And only then can the biomass bring some small improvement. So then we decided to have a deeper look at the exchange rates, and we wanted to study the error propagation from metabolite concentrations to calculation of exchange rates, and then to FBA predictions of growth rate. And we wanted to do this because CHO medium is very complex, so this is just for illustration of what components it has. And typically, to run FBA, we need to measure more than 20 metabolites. So these include, for example, glucose, lactate, and all the amino acids. And we wanted to see which metabolites have the biggest impact on the predictions. So to do this, we simulated metabolite concentrations over a batch culture, and we simulated data with different errors, so between 2 to 20% error, and with sampling every 6 hours, 12 hours, or 24 hours. And we did this for 23 metabolites. Then we calculated rates and errors by fitting an exponential function, and we did this 1,000 times. And the most important finding we had was that lower rates have higher errors of the fits because the change of concentration is very low in these cases, and then it's much more sensitive to experimental errors, and it often happens that you even get a fit that goes in a wrong direction. So then we took these rates and used them as input for FBA, and here we can see the results. So these are predicted divided by the reference growth rates, and this is the error of the metabolite concentrations. So as you can see, the bigger the error of the concentrations, the bigger is the error of the predicted growth rates, but the error kind of amplifies. So for example, at 20% error of concentrations, which does not seem that bad, we get up to 350% error in the predictions. So we overestimate the growth by 3.5 times, or we also have a lot of cases where we predict no growth at all. And also more than half of the solutions actually become infeasible. So to obtain good predictions, we really need to take care to have accurate metabolite measurements and also high sampling frequency. So on the left, we have sampling every 6 hours, and right is 24 hours. And we also saw that the biggest impact have the essential amino acids with low uptake rates, and this set of amino acids is cell line and condition specific. So next, on the previous slides, you saw that iCHO can predict growth rates very accurately if you provide it with good experimental data. But the next question was, can it also predict intracellular fluxes? Because in the end, this is what we need to improve the cell lines. So to find out, we took some literature data sets. So we took 20 data sets with 13C metabolic flux data, and we compared these fluxes with predictions by pFBA. So here you can see the predicted versus experimental fluxes. And it shows that most fluxes are underestimated in the pFBA predictions, and that the worst predictions were obtained for the TCA cycle. So this suggested that there is some problem with the energy metabolism. For example, there could be insufficient energy demand. So one idea that came was that it could be the missing non-growth associated maintenance energy, because currently the model doesn't contain any value, and there are also no values available for CHO in literature. So we decided to estimate the mATP, and to do this, we scanned a range of different mATP values, and we looked at the agreement between experimental and predicted fluxes. So this shows the median relative error. And then we've always found a value where the median error was the lowest. So in this case, it was around 5.4 point something millimole per gram dry weight per hour. And we did this for all 20 datasets with 2 biomass equations. And here you can see the results. So on average, it was around 6, but the range is quite big. And then we used these values as a constraint for the FBA predictions. And you can see that basically with just one constraint, we can significantly improve the predictions of fluxes. And it looks really nice. Now, the only pathway that is not predicted very well is the pentose phosphate pathway, but we are still looking into this. Then we also determined the mATP experimentally. And we did this by running continuous fermentation at different growth rates and measuring the exchange rates. Then we calculated energy consumption and extrapolated to 0. So the intercept here represents the maintenance energy. And compared to the computational estimate, we got a bit lower value, but I think they are still quite comparable given the experimental uncertainty. So to summarize, in this work, we measured biomass composition, exchange rates, and maintenance energy for different CHO cell lines and conditions. And we found out that the biomass composition is very variable, except for amino acid composition, but it only has small impact on growth rate predictions. Exchange rates have bigger impact, but to get accurate rates, we need to we need to do high sampling frequency, and we need very high accuracy of the measurements. And the most important rates were the low uptake essential amino acids. And then finally, maintenance energy was important for intracellular flux predictions, and we obtained values between 4 to 6.4 millimole per gram weight per hour. And if you want to learn more details, you can check out our publications. And finally, I would like to thank my supervisors and colleagues from Univie, ATSIP, and Boku, our company partners Novartis and Biocrates, PhD program Biotop, and our collaborators from other groups from all over the world. And thank you for your attention.